feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us. Professor Christiaan Leeuwenburgh is our guest tonight, and he will talk about iron deregulation and, of course, aging. This will trigger lots of questions, I assume, so please send them and your remarks, your comments using the Q&A function. We are starting, as always, with a short video today given by Ewan, our research assistant at the National University of Singapore, and he will talk about the ABLE study, how to select individuals who are biologically older but healthy.
Thank you, Prof, for the kind introduction. Hello, everyone. As mentioned, my name is Ewan. Today, I will talk about the selection of the biologically older, healthy individuals from the screening phase of the ABLE clinical trial. Project ABLE is a double-blinded, placebo-controlled longitudinal intervention study in middle-aged healthy participants. The study aims to investigate the geroprotective effect of the investigational product, calcium alpha-ketoglutarate, on biomarkers of aging. The total duration of participants in the study is 9 months, with 6-month interventions and 3 months follow-up. So why pre-screening and screening phase are needed? There's 4 aspects, which are homogeneity of study population, which helps to reduce variability in the data, minimizing the confounding factors which can distort the relationship between the interventions and the observed outcome. For safety purpose, it helps to identify individuals who may be at a high risk of adverse events or complication due to their medical history. Eventually, it's about the data quality. We will have a reliable and accurate data. Firstly, the potential participants filled the application form. Those aged between 40 to 60 years old and did not have the following chronic disease in the table were invited to the screening visits. During the screening visits, after signing the informed consent, the blood was drawn for clinical laboratory assessment and DNA methylation age analysis. We screened their eligibility based on the following inclusion and exclusion criteria of the study. The inclusion criteria is DNA methylation age, which was measured by the mean of the 4 clocks: Howard, Hannum, PhenoAge, and Grimmage. Participant was considered if the DNA methylation age is older than their chronological age. This is because we are investigating whether calcium AKG able to reduce the biological age by the end of the interventions. Participant was excluded if they met one or more of these exclusion criteria. This figure represents the distributions of DNA methylation age deviations, which is defined as the difference in biological and chronological age in the ABLE screening participants. Close to 20% of the participants were biologically younger by a mean of 3.7 years, and 80% of the participants were biologically older by a mean of 6.8 years. All participants were also assessed for the screening exclusion criteria. As just discussed, we have around 56% of the total population who had not met any exclusion criteria. 30% of them have met one exclusion criteria, and we only have 0.4%, which is one participant, who have met 4 exclusion criteria. Further dive down, 44% of the biologically older participants did not met any exclusion criteria. 23% of them met one exclusion criteria, 10% met 2, 2% met 3, 0.4% met 4. Interestingly, there were 7% biologically younger participants who met one exclusion criteria and 1% met 2 exclusion criteria. This is the frequency of individual exclusion criteria met by able screening participants. Almost 48% did not meet exclusion criteria, whereas the blood pressure and waist circumference were the 2 most frequent criteria, with 27% of the participants having elevated blood pressure and 70% had increased waist circumference. And now, the screening process has been completed. I would like to thank to this team who have been working tirelessly on this project. Currently, the ABLE study is still going on, and we're looking forward to share the results of the study in the near future.
Thank you so much, Ewan. It's always a pleasure to see how we could select our individuals. It's my pleasure now to introduce Dr. Christiaan Leeuwenburgh. He is a professor of physiology and aging at the University of Florida in the United States. His studies really focus on the molecular mechanisms of mitochondrial dysfunction, iron transport deregulation, autophagy, and oxidative damage to our DNA, which we have in our mitochondria in not only animal models, but also in humans. And his findings also translate not only from basic sciences, they are now translating into clinical trials with the aim to improve human health span. Christiaan, it's a pleasure to meet you. You are going to talk about the hallmarks of aging and really focusing on the iron deregulation in aging. The floor is yours.
Thank you very much. I really appreciate the invitation by the Center of Longevity. So I'll talk about iron. So let's start quickly to review why iron is essential for life. You can see here on the right top that iron binds to oxygen. And we've evolved around iron and oxygen and the ability to transport oxygen to tissues in this way. Oxygen is a diradical. Iron has radical properties, but they can bind and transport oxygen to the mitochondria. Here, also iron is essential for life. Iron is incorporated into the electron transport chain complexes, as you can see, and is also important for the dance of electrons. Electrons can bind to iron very moderately and then be transported, and this helps create a proton gradient and therefore produces energy. So you can see iron is essential for life, but we'll see that iron might become damaging over time in very old individuals. We all know this, aging is progressive. We know what fitness means. It's being, not only physical, but cognitive healthy. In biology, it means the ability to survive and reproduce in an environment. And you'll see at the end, we'll talk about, health span and how we can improve health span and potentially even iron homeostasis with some, practical interventions. We'll talk about that. But you can see, very healthy generations of females here, and they've aged very well. We know aging is characterized by a decline in memory, to simplify, and there's also plenty of evidence that iron accumulates in the brain. What's maybe less studied is iron accumulation in other tissues. Muscle is easily accessible in humans, so we've made an attempt to characterize iron accumulation in muscle. What you can see in this slide is that as we age, also we slow down. Our walking speed declines. And that's one parameter we can measure in humans. And keep in mind that the way we function is also predictive of disease and mortality. So you'll see data on 70 to 80-year-olds. Some are high functioning, and they have a different trajectory to mortality and disease compared to low functioning, which will go easier and quicker into disability. So there is something very different between these individuals, these groups of people, and we're trying to study that as well. Here are the hallmarks of aging. Obviously, it will take too long to go over all the hallmarks. We went from 10 to 12 now, and but I'll indicate a little bit where iron might be important, and also where we might be able to intervene to have a better homeostasis of iron during our lifespan. We'll talk about genomic instability, and we know with aging, there is an increase in mutation deletions as we age, and even telomere attrition might be contributed to iron damage over time. We already indicated to you that the mitochondria is a source where iron is needed, and we'll see that iron transport into the mitochondria and handling will be an issue. Now, we'll also talk about autophagy and the loss of proteostasis, because we can intervene here and possibly control some of the iron accumulation that occurs with age. Here is, a simplified graph from the paper Hallmarks of Aging, an Expanding Universe. And, it's hard to say where aging starts, but I still would like to think that nuclear aging is very critical to life. And we certainly know that exogenous DNA damage, due to smoking, toxins, et cetera, can cause accelerated aging. But also there's endogenous DNA damage. People that don't smoke, they don't get exposed to a lot of toxins. They still show genome instability. Certainly in a model system, way back in 2005, we established that large-scale mutations, and later on also others have shown deletions are important and accelerate the aging process. Certainly keep in mind that your DNA repair pathways are very important and part of this formula, so I'm not ignoring that, and there's some changes in that as well, but endogenous DNA damage has to come from somewhere, and it has to be still likely reactive oxygen species, specifically hydroxyl radicals. So this slide just tells you that in many human animal tissues, it's been found that iron accumulates. And again, the message is as iron, as an essential mineral, iron is a double-edged sword. Without iron, life is not sustainable. And with age, many organs and organisms exhibit age-related cellular iron accumulations. There's genetic conditions in humans, such as hemochromatosis, where iron accumulation is detrimental. Now, where does the autophagy, where does the autophagy fits in, and what are some of the studies we're conducting to stimulate autophagy? I'll explain autophagy real briefly here, and then I'll show you in the context of iron control and removal within the cell. So most of you are familiar with autophagy. It's broadly nuclear regulated. You see an expansion, kind of the simplified version is a small garbage bag being formed, and then you see cargo taken in, such as damaged mitochondria and aggregate proteins, and they're recycled and made back into sugars, amino acid, fatty acid, nucleotides. So this is referred to as macroautophagy, and very important in a variety of turnover mechanisms in the cell. It's very important to keep your proteins new and happy and turning over. To get back to the iron story and then autophagy, and this is what we're studying in older adults, there's an accumulation of iron as non-heme iron, so not the functional heme iron as I showed you earlier, like in hemoglobin or within the electron transport chain complexes, but there's this storage iron that increases in the tissues with age. And it's important that this is turned over by autophagy, of course, in conjunction with the lysosome, and then turn into useful functional iron pool, which can help mitochondrial function and the synthesis of heme enzymes in the mitochondria. Also, the proteosome is able to help with this process and turn over ferritin. And at the end, we'll talk a little bit about time-restricted eating, caloric restriction, and we have a hypothesis that's probably the only way right now that we can help to turn over iron in the cell. Again, a complex slide, but the bottom line of this slide is we take in daily iron, 10 to 20 milligram, and only a few milligrams are actually absorbed in the gut and then transported by transferrin. Most goes to your bone marrow where you make your red blood cells. And then, the iron also is transported to tissues and stored in ferritin. Could be muscle tissue, really any tissue, and the mitochondria has ferritin storage. But the general problem is that we have no physiological excretion mechanism. So the only way to lose iron on a day-to-day basis, the 1 to 2 milligrams to stay stable, is to lose skin cells, epithelial cells, or from blood loss. So it's very important to, of course, regulate it, through your diet, but also within the cells and the systemic system. This slide just shows you the master hormone hepcidin, how it works. So if plasma and tissue iron level increases, this hormone hepcidin, which will be discussed in the older individuals, it will be released. It goes into the bloodstream, and it will degrade ferroportin, for example, in the gut, and thereby it can control the levels of iron, and iron levels, again, they go back down to a normal level. So this hormone is very important to regulate plasma levels of iron, but also iron export out of cells, such as muscle cells, which is depicted here. So with age, it's now well established that there is a significant amount of inflammation. Here are some inflammatory cells, but also senescent cells can produce cytokines. And IL-6 is one of them, but the liver where hepcidin is produced is susceptible to increased hepcidin levels. So more inflammation, more hepcidin, and that's where the issues start. It starts breaking down ferroportin at different places in the gut, in macrophages, in muscle. So transport is already affected due to the chronic inflammation over time. Going back to free radical biology and where iron in high levels can be harmful is where you can see it, the circle in reaction number 7 and 8. But before reaction 7 and 8, I do want to point out that equation 3, 4, 5, and 6, for example, are enzymatically regulated and they produce peroxide, which is a relatively stable reactive oxygen species. But the peroxides, in the presence of redox active iron, ferrous iron, can be split into a hydroxyl radical, for example, and this doesn't have an enzymatic system. So again, peroxides are well controlled, but phantom reactions produce hydroxyl radicals, and I'll show you some evidence of that. And many have published many papers, including, for example, Barry Hollowell, who's taught me a lot in free radical biology, and these are some of the schemes. So there's no defense against this hydroxyl radical, and really, as far as chemistry, it's one of the few things that can originate from this ferrous iron. What you can see here is, DNA and RNA, and you see the hydroxyl radical in the center, and then you see different structures created that, certainly they can be removed and repaired, but over time, the systems don't keep up. You can detect these altered DNA and RNA bases in the urine, and we've done that in the past, and others have done this. So yes, we are removing this, but with time, things can accumulate and become more intense. So the premise we're working with, we and others have shown increases in ferritin, the non-heme iron pool in tissues. Again, this can lead to iron overload and ROS formation, and can cause cytosolic damage, but also damage in the mitochondria, where the same could find place. And this is a source of reactive oxygen species. And to make it maybe more stronger is we lose a lot of mitochondrial DNA copies. 50% of our DNA is lost over time. So, this can affect function. We're lucky to have multiple copies. But what we'll see is that, hepcidin can affect the ferroportin, the export of iron out of the cell. So this is impeded. But maybe to a surprise, these older individuals also have issues with import of iron. And the major way to import iron is to the transferrin receptor, and it's important to bring in new useful iron to be internalized and to be shuttled to different places like the mitochondria. So you have this double whammy when you're very old, you're accumulating, you can't get it out, but you can't even get new iron in. It's well established in cell cultures. When there's a lot of iron in the cell, it actually shuts down import. So let's go to some human data. So I'll show you a few pilots, and then we have a larger clinical study ongoing. But basically, recruit older participants. We take muscle biopsies. We take blood. We look at functional performance. We do a variety of measurements, including mitochondrial respirometry. And then we do functional tests. Well, we do a lot more. We do sleep, we do mobility. You'll see it a little later on, but there's a lot of tests that are being conducted, cognitive tests as well. This is an important slide that shows you both the inclusion-exclusion criteria. And I want to focus again on the fact that we're recruiting relatively healthy individuals between the age of 70, 80. We do have in the pilot study some young controls. They're low functioning and high functioning based on their walking speed and other, classic geriatric tests. They have a sedentary lifestyle, so, exercises are not allowed to be in the study. They're normally moving people. We want to take the confounder of exercise out of the design, and they can walk to the clinic, ability to walk, and they have no cognitive impairments. On the right, again, notice that anyone with a disease condition is excluded from these studies. We can go over them, but you can see these are major conditions, including any type of inflammatory condition, because we saw that, hepcidin can affect the level of iron import and export in the gut and into different tissues. So what do we see here? This is hepcidin level. Again, hepcidin keep in mind, not only in the gut, but also in other tissues, can break down ferroportin. And you see the highest levels in the low-functioning individuals in this pilot study of about 30 in each of these groups. What is also interesting, besides ferritin, we can also with mass spec detect different isotopes, and the largest level of isotopes, and I know the scale is in a negative sense because it's quantified dependent on the total iron level, but it indicates that there's an increasing pool of labile iron in the cells with this iron overload. So it's also the different isomers that we can test with ICP-MS that show that those low individuals have very reactive iron. So next, I just want to show briefly, and then we'll get to the interventions, some of the mitochondrial proteins that are important for import and control of mitoferrin and control of iron in the mitochondria. Actually, first, I want to show you, and I already indicated this, the transferrin receptor. Again, this is the major import system of iron into the cell. And look how remarkable low the low-functioning older adults are. They have very little transferrin, again, despite having large levels of ferritin in tissues. We also looked at other proteins, CYP8 and CYP14. Those are known as zinc transporters, but they also have a moderate ability of iron transport. And a similar trend is there, but it's most striking with the transferrin receptor, which is the major import system of iron. Here, I'll show you the data on mitoferrin and frataxin. What is interesting is in the low-functioning individuals, mitoferrin is markedly increased, possibly as a adaptive mechanism, which you can see it's very much deregulated from the young and also different from the high-functioning. Maybe more important is the frataxin level. Again, frataxin is very important for the assembly of the iron-sulfur proteins. It's an iron chaperone and helps with iron storage. And we know, for example, with genetic conditions like Friedreich ataxia, this causes issues with walking in those individuals with very low frataxin levels. So, frataxin might be a target for, gene therapy in the future on these individuals to at least try to stabilize some of the iron that is now roaming free and increasing free radical stress. So this is a large study that has started. I won't be able to show you data, but I'll just explain the design. It's the same inclusion-exclusion criteria, but no young control is included because this is a longitudinal study. We will be able to take muscle biopsies and blood at baseline, but also treat, and maybe even longer, 5 or 6 years if we get a renewal, and we can follow iron homeostasis over time. So that really the comparison is between the low and the high functioning. And I just want to stress in this, in this table that the main difference in these people are their walking speed. We also looked at grip strength. So, functional measures are different, not their disease state, not their BMI, same age. We're looking at their function to distinguish their path and their trajectory. So the design is depicted here. You can see there's a screening visit, then there's baseline, where a variety of tests are done, and then there is a muscle biopsy, and we also look at mutations and deletions. In some omics analyses will be performed. So you can quickly get a grasp of the scheme. It's very intense, but we're doing well. We've got about 90 people recruited. Okay, so this is the fun part, hopefully, where we can intervene. I'll be quickly in 5 or 6 slides to stay within the time. But what we do know is that with an acute injury and some select diseases, there is the ability to chelate iron that becomes free. But, chronic aging, you can't give chelating compounds. So acutely, iron chelators could be of interest in conditions where there's tissue damage in the heart. You need to be quick in the kidney, the heart, ischemia, reperfusion, stroke. Maybe some mild compounds can be mild chelators, but aging is much more challenging. We need to affect all these processes: absorption, transport, import, export. So they all need to be improved. So one potential way, and I'll show you some data related to iron, is to do time-restricted periods of time-restricted feeding. So there's episodes of periodic or literal or no caloric consumption. There's different regimens, and everybody is doing this. Everybody has slightly different regimens, but the most important part in these regimens from basic science studies is that there needs to be a fasting period. And this paper of not our group, a different group in Nature Metabolism, shows that the most important factor is these fasting periods in these animals. And if you see the top mouse here, there's restriction of calories without fasting. They still turn old, but in the other models they studied, is restriction of calories or fasting alone will keep them healthy and fit, and daily fasting without caloric restriction is also beneficial. So I'll show you studies we've conducted on caloric restricted animals, but what we know in these caloric restricted animals, we feed them once a day in the morning. So they're really also time-restricted eating for a period of 22 to 23 hours. They eat their food very rapidly. So although the benefits of all the caloric restriction studies is thought to be due to reduction in calorie intake, CR studies in rodents often inadvertently impose an extended fasting time, since the animals are typically fed once a day and thus consume their calories in a short period of time. So we know some data in mostly younger, healthy individuals that this time-restricted eating can have some benefits, but we don't know much about very old people if we can do that. Let me show you a little data related to iron accumulation. You can see here there's some animal data, 8, 18, 29, and 37 months. So very old animals. And here's the non-heme iron amount. You can see in the muscle it increases in the normal ad-lib fat animals, but in the caloric restricted, time-restricted animals, fat, it kind of stabilizes. It doesn't accumulate over time. And the liver, which is a major storage of non-heme iron, also, there's probably better turnover. And I'll show you some autophagy slides and, potentially evidence of iron removal. Here is some of that. And we know that lipofuscin is, kind of a biomarker of aging. It's an iron lipid deposit. And what you can see very clearly if you look at the very old animals, these are peripheral nerves isolated from the muscle, by the way. You can see accumulation of lipofuscin in these very, red spots. And this is much greater than an 18-month, animal. So there's a marked increase, but you can see in the caloric restricted, time-restricted fat animals, there's a markedly less accumulation of lipofuscin. Also, to go back to the proteosome system, what you can see in peripheral nerves, and we've done this in other tissues as well, you can see darker bands as the animals get older. And these are ubiquitinated proteins. So these are proteins getting ready to be removed by either the proteosome system. It could be macroautophagy as well, but you can see with the caloric-restricted, time-restricted fed animals, there's less of this accumulation occurring. And on the right is the quantification of this. In addition, what we showed is that it appears that in these peripheral nerves, that the speed of removal is not very efficient in the older animals, but it's more efficient in removing the garbage that occurs with time. So again, we have right now we have some pilot studies ongoing on time-restricted feeding, but we also have a larger clinical trial on the review where we want to stimulate these processes, and we would use a 16-period fast in those studies in older adults, and the pilot study showed that this is feasible. So in summary, tissue non-heme and labile iron increases at old age. Also in the mitochondria, I didn't have time to show you, there's an accumulation of iron, and those mitochondria are more prone, that those old mitochondria that are accumulating this non-heme iron are also more prone to releasing pro-apoptotic factors. And, ferroptosis is part of this, is kind of a general name where, iron is very much so implicated in mitochondrial dysfunction. A lot of big papers on that topic. We saw that mechanisms of iron uptake, transferrin, ferroportin, ferritin are deranged in aged tissues. And we saw, clearly that these low-functioning older adults, there was even a worse outcome in those biomarkers. Certainly, iron is not going to be everything, but again, it's one aspect we want to study, and we're going to do this in a longitudinal study. We are doing this right now. We've got 90 participants. The goal is 120, and we hope to follow them for many years. Right now, the plan is for a period of 5 years. We also believe that right now, preventing obesity, because obesity is also associated with iron deregulation, but time-restricted eating might be a way. Exercise has shown to provide some benefits, and that's a way possibly to normalize iron. Again, I want to thank you, and I'm welcoming questions, and many investigators have contributed to this work. I'm very grateful for that.
Thank you so much, Christiaan. Wonderful presentation, really focusing on iron. I learned a lot. I must say, as a clinician, I very often prescribe the iron, and I think now I am thinking a little bit different about it. But maybe we can also go towards clinical implications in the end of this discussion. But let me start with your clinical studies. It's very nice seeing that you are really moving into human studies, longitudinal observational studies, intervention studies. You're focusing hugely on muscle. I understand, because this is accessible, you can biopsy it. What kind of organ, if you would target humans, would you like to biopsy if everything would be ethical?
Yeah, now there are some more sophisticated brain MRI imaging possible, so that would be not as invasive. Now we're getting blood. We are taking, in some individuals, we do get a little more fat, and but yeah, hypothetical, the liver would be nice because it's a large storage area of iron. Coming back to your comment, you are still doing the right thing of giving. Sometimes, in some cases, you're manipulating iron because, again, they need it. Sure, there might be accumulation of iron. We're not talking so much about anemia, we could go there, but still at the same time, import is also affected. So you need iron to go in. It's just the general trend we're seeing is this storage and turnover is really affected. What I didn't make really clear is autophagy slows down in this presentation. We and many others, I think we were some of the first to show in animal models, for example, during ischemia reperfusion in the liver, that if autophagy doesn't work and we were able to do transfections, if autophagy doesn't work, it cannot remove the damaged mitochondria. And I didn't make clear that with aging, autophagy is really failing as well.
Yeah, absolutely. So you might then, from a intervention perspective, say, okay, we have to fix autophagy rather than iron up the iron.
Yeah, it's both. I mean, a condition, for example, I'm jumping around a little bit, cancer cachexia. There's large levels of inflammation. So there's also a marked reduction of this transferrin receptor. Those, in this case, animals and some small human studies, again, they benefit from iron supplementation because they still, at the end of the day, you're going to need iron to go in and to help with energy production. And that's what they see.
Yeah. So you already touched on autophagy. You also had a slide where I think the ones who are studying inflammation and Claudio Franceschi's inflammation theory might be sitting upright, seeing that inflammation really drives, you say, causally, the hepcidin levels, and that is associated, if I understood correctly in your studies, with frailty. So should we, or do you know of studies actually testing if maybe not IL-6 and CRP, which are all markers for inflammation, but maybe hepcidin is a much, much better predictor or associated with frailty? So the incidence of frailty as a predictor, but also in terms of associations, much, much better than a walking speed or a hand grip strength.
Yeah, it's a tricky thing. I kind of made the point, I thought, aging starts from the nucleus. But, it's a variety of things. Inflammation is just hugely important. Now, to twist it back to mitochondria, if they become damaged, it's never easy to say where things start. If they become damaged, they can release mitochondrial DNA. And again, anything that's within the mitochondria, cytochrome C is another example, mitochondrial DNA goes out, it can stimulate inflammation. And people are interested to look at this in the circulation as well. So, what is a better predictor is sometimes challenging to say because, a lot of things happen with time.
Yeah. But isn't that in our field? And while being so honored having many guests here talking about hallmarks of aging, in these 12 hallmarks, which were 10 or 9 or 10 previously, what is the chicken and the egg? And of course, in the paper you cited, there is a sort of one leads to the other. But what do we actually know, and would you actually say iron deregulation needs to be a hallmark? Or is it so much downstream and the hallmarks are really the cause of the iron deregulation?
No, it's challenged. They all interact. I mean, there was a paper recently, also in Cell Metabolism, I think it was Nature Aging, that found that iron accumulation stimulates senescence. I'll gladly send you that paper. It was a nice paper. Again, if senescence is everything, which some people believe now or think now, which I'm still skeptical how many senescent cells we have as we are aging normally. I'm not talking diabetes or heart disease. There might be a significant increase, but relatively healthy individual, how many senescent cells we have. But again, what's interesting, senescence is one of the top candidates for drug therapy with senolytics. That a recent paper showed that iron drives fibrosis and senescence. And, one of the other hallmarks that's not on there is fibrosis. You think about fibrosis and old age, I'm getting there, stiffness. And it's a big biological process that's changing over time. And fibrosis could be one of the hallmarks of aging. And maybe it will be added in the future if we can, intervene there in animal models.
Yeah, I think let's do not touch how many hallmarks we have. I think we all have our favorite one. I didn't know, and that was depicted in the first slide, that we are taking 10 to 20 milligrams of iron each day, and only 1 to 2 milligrams are actually absorbed. Do you know why? Why is it, I would say, why don't we use the iron we have in food?
Yeah, well, it's protective. I think we've evolved with very little iron during evolution. And so anytime, maybe, humans way back, they had less iron intake. I wouldn't know. But it's a great point, but it tells you that we only want to have 1 or 2 in the condition of hemochromatosis. A normal human has about 3 grams of iron in the body, but in those people, it's increased by tenfold. I've got some slides on it, but I won't move around. But yeah, and they have early death, early disease at age 20. So yeah, it's very, very well controlled. And with people with hemochromatosis, there's an issue with the absorption, and iron just keeps flowing in, and they can't get it out because there's very limited ways to get it out. But yeah, great point.
Yeah, and that brings me, before going into the intermittent fasting section you had, to if I then as a clinician give iron tablets, I'm not talking about the IV infusions, but the tablets, and receptors, the transferrin receptor is actually down. How do I get the iron in? Isn't that the problem? Shouldn't we do something about it?
I tell people with any supplements, there's all these steps to get it at the right places. If all these steps are synthetic pathways, I'm not going to throw some other pathways under the bus, but there's a huge market for, and we do have a clinical trial coming out in Nature this week, Nature Communications, on boosting NAD levels, and there's NR, NMN. You can supplement now. You can buy it over the counter. But if your steps of synthesis, if there are issues with that, you might never get to NAD. So your point is well taken with iron supplements. If we don't fix the steps for import export in that case, it's an issue, but maybe a little bit will trickle in. But at the same time, there could be some toxicity. But do you want to stay alive with some damage? Yes, there's a risk and benefit there.
Yeah, and in the end, obstipation. But let's move into another field. You really touched very nicely on intermittent fasting, time-restricted eating. That's interesting. We see, of course, benefits in the animal models, at least, on many organ systems, while not at all, but many of them, while having a regime of intermittent fasting. Do you in your studies actually look at the intake? So is it the total amount of iron, or is it just the timing of the iron? And if it's the timing, how can that be? Because we just talked about the receptors getting it in. Are they those sensitive of when they need to be active?
Yeah, no, it's a great point. And again, in the longitudinal study, we're monitoring their food intake. We all know that this is not easy. We have an extensive survey. In this case, we're not going to be able to feed those people and control over a 5-year period of time. We're working with Tufts University, some expert in nutrition there. We're also monitoring sleep. We're monitoring activity levels. But yeah, we're going to get the data on those low and high functioning, and there will be covariates to assure that they have a similar dietary intake.
Yeah. Yeah. So maybe we should also look at meat lovers and spinach lovers versus the vegetarians or vegans not eating spinach or beetroot.
Well, that's a great point, because with somebody else, Alex Young, he studies heme iron intake and non-heme iron intake. So again, heme iron is mainly through meats, and non-heme is more plant-based, and probably less, and there's less concentration of iron there. And he studies the incidence of cancer. So he has also large data sets called One Florida, where we have iron levels in patients of millions of people, and then he looks at iron consumption, and then also at the iron markers, and then he looks at prediction of different cancers. Again, an association, but it's important to look at that if it matters. We do know that high intakes of meat is associated with, for example, with cardiovascular disease and also cardiac iron accumulation.
Yes, absolutely. I see that we have many questions from the audience. I will hand over to Dong Leu, our PhD student at the Center for Health Longevity. Go for it.
Thank you for the introduction, Andrea, and thanks very much, Prof Christiaan, for the very interesting talk on iron homeostasis and aging. I also learned a lot of things from you. Now I'm going to ask the questions from the audience. It seems like, first of all, many of our audience are interested in the aging-related changes in human iron homeostasis, or in your words, iron dyshomeostasis. Do all tissues accumulate iron with age? And is free iron instead of total iron the problem?
Yeah, so that's a great point. Many tissues have been studied. So as far as I'm aware of, we would think, most tissues, but not all tissues have been studied, but most have been studied. Iron accumulates. Humans, it's a little more challenging, and muscle is probably one of the easiest accessible tissues. And, brain imaging has certainly been done. So what was the second part of that?
This audience is asking, is free iron instead of total iron the problem?
This is a great question and a great challenge for science. Because, again, ferritin goes up. Ferritin is very protective because it binds iron. So it goes up for a reason. So it's adapting to the iron overload. So at one part, it's protective, but maybe during aging and small injuries and the autophagy not working, the susceptibility of the ferritin to release free iron is increased. Certainly with an real acute fall, or but it's going to be. But it's very challenging. So we have isotope techniques. I showed some. We do histochemistry, where you can get a feel of the free iron. But the technology, hopefully in the next few years, will get even better with mass spec, where you can do specific histochemistry, and you can do mass spec on histochemistry. Most of you young people can do all that. Yeah, and then we can detect and localize iron into the cell. And we need help from basic science there as well.
I see. That's the reason why you're trying to give advice on giving different, like the slow releasing, like the fast chelator for iron. And we really need to reach a nice balance for iron. So let's go to the next question, which might be related, because you earlier on showed that iron also accumulates in the brain and mentioned about the use of MRI images. Is there any current evidence on association or causation of iron accumulation or intake and aging-related, like neurological conditions?
Yeah, the most evidence is in macrophages. And macrophages, in general are, really they're the good guys, and they clean up, they remove things, and they're specific in the brain. But as they get older, there's evidence they're accumulating iron as well. Is that causal? It's tricky to establish, but it's associated with nerve damage and different neurodegenerative diseases. So yeah, the biggest evidence is through macrophages that are being detected and stain and autophagy or in biopsy samples. Yeah.
Oh, I see. Thank you for your answer. So let's go to the next question. So this audience is asking about hepcidin. So he was asking, do the levels and activity of hepcidin change with age? Besides that, inflammation affects it.
Yeah. So, as you saw on the slide, there's a change, maybe 30, 40%. Again, you might think that, first hepcidin is it's a very important hormone. It's there for a reason. So if you have an infection acutely, we want iron levels actually to drop rapidly. And this will kill the microorganisms that are infiltrating your system. So during normal, an infection, it's important this hepcidin goes up acutely, and it's kind of an acute hormone synthesized from the liver, and the liver can do this very rapidly. But again, with aging, it's this chronic, slow, constant release, and it doesn't turn off. For an infection, definitely in young people, it turns back on, and then it turns back off. And some older people are probably better in turning things on and then off. They're more resilient. But yeah, this long-term chronic hepcidin is causing an issue.
I see. So maybe in the future, there's a way to target hepcidin too.
Exactly, and I forgot to mention that. Yes. So yeah, people are interested in that. And they've done this on IL-6 a lot of IL-6 antibodies or compounds, but the success has not been great, which again, unfortunately shows you that it's complex and there's probably multiple factors. But yeah, one target could be hepcidin, correct.
Okay. Thank you very much. It's really thought-provoking. We are going to the next question that really triggers my curiosity too. So this person is asking, is proteosome activity itself impacted by iron accumulation? And would calorie restriction help to prevent the impairment?
Yeah, so we have done experiments with caloric restriction, which is time-restricted eating, either short-term or long-term, and proteosome activity is up. So, whether iron, the question, has a negative effect directly on proteosome activity, I don't know, but we know if there's a lot of aggregate, ubiquitinated proteins, the proteosome, probably can't handle that, and they just don't have the capacity to affect that, affect the removal. And yes, a large amount of aggregated proteins can affect proteosomic activity. Correct. That is correct. Yeah.
Okay. Thanks for the answer. Early on, we were talking about autophagy and iron accumulation and also aging. So this person was asking, would promoting specifically mitophagy be effective in reducing iron accumulation, given that it enriches in mitochondria and so on?
Yeah, again, it's a good point. I think, yes, to turn over, any protein, but certainly organelles like the mitochondria are critically important, and recycling there can release nutrients, I'll say, including iron, for recycling and incorporation into new mitochondria by mitochondrial biogenesis. And it could be one, healthy mitochondria, again, through exercise. Time-restricted eating will also turn them over. So that could be one way to improve iron homeostasis as well. Great point.
So there are actually a lot of approaches to
Yes.
modulate the iron homeostasis.
Yeah.
Okay. I got the cue that we have limited time, but we really have a lot of questions. So one last question that I'm sure many of our audience are interested, when we're discussing different diet intake of iron in clinical trials, it's interesting to think of ourselves. What suggestion would you give for like mid-age group people or elderly on their iron intake in their daily life?
Yeah, I think, we do know the evidence of, too much, meat intake. It's very important, but it's very high in iron levels. So I think that would be probably an easy way. I'm not a vegetarian. I love meat. I like meat. But I think, in moderation, seafood is not as dense in iron. So yeah, and taking in an increased amount of plant-based non-heme iron would be probably beneficial at any age. Yeah.
Okay. Lifestyle advice taken.
Yeah.
Thank you, Prof Christiaan.
Thank you very much, and the audience. Yeah.
Thank you so much, especially for the advice. I'm compensating for you both. What I learned is iron is essential for life, but it's really about the balance. Thank you for joining today as our audience. Please leave all the comments, your suggestions in the chat box using the panelist and all attendees options. It's very valuable for us. We will close the webinar with evaluating how nursing students can influence a person's life. Take care.
Students in Drexel's accelerated nursing program spend a stint in Folkways, a retirement community in Lower Gwinnett.
I think it made me realize how much I actually enjoy working with this population. You guys climb the Eiffel Tower. Listening to their stories and their histories and things that they've accomplished is just so amazing. Anything else you want?
I lost my mom to pancreatic cancer. She had these nurses. She liked learning about their lives, playing cards with them. If I can kind of pay that forward and do that for other people going through similar things, would mean like the world to me.
The student nurses make a big difference in the life of residents.
Oh, I think it's wonderful. Nothing like having young people in a place like this.
They bring me tomatoes, they bring me chocolate ice cream. They've been very helpful, not only with gifts, but a morale boost.
And for students, the experience can be life-changing.
Whether it's remembering that they really like chocolate pudding instead of vanilla pudding, or even, remembering how they like to do their hair. Just seeing the satisfaction and the happiness on their faces is definitely very rewarding.
When I leave here at the end of my day, I feel like I've made a difference in the residents' lives. I put a smile on their faces, and it makes me feel good to walk out the door.
Tamala Edwards, Channel 6 Action News.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.